Hello viewers, welcome to the lectures on UGC model syllabus in zoology. Today's topics are polymerase chain reaction and gene libraries. Polymerase chain reaction or PCR is an in vitro technique used to synthesize large quantities of specific nucleotide sequences from small amounts of DNA. It employs oligonucleotide primers complementary to specific sequences in the target gene and special heat stable DNA polymerases. The first step is to synthesize DNA fragments or primers with sequences identical to those flanking the targeted sequence. These synthetic oligonucleotides are usually about 20 nucleotides long. The reaction mixture contains the target DNA, often copies of an entire genome, forward and reverse primers, a thermostable DNA polymerase, and each of the 4 deoxyribonucleoside triphosphates or DNTPs. PCR requires a series of repeated reactions called cycles. Each cycle has 3 steps that are precisely executed in a machine called thermocycler. In the first step, the target DNA containing the sequence to be amplified is heat denatured at 90 degrees centigrade to make it single stranded. Next, the temperature is lowered so that the primers can anneal to the DNA on both sides of the target sequence. Because the primers are very small and are present in excess, the targeted DNA strands anneal to the primers rather than to each other. In the third step, DNA polymerase extends the primers and synthesizes copies of the target DNA sequence using DNTPs. Only thermostable polymerases can be used in PCR technique, as the synthesis of complementary strand is carried out at about 70 degrees centigrade to increase efficiency of amplifying the target sequence. Lower temperatures can allow non-specific binding of probes and amplification of non-target sequences. If a heat labile DNA polymerase is used, it would have to be replaced after each denaturation or heating step. Two commonly used heat stable DNA polymerase are Taq polymerase from a thermophilic bacterium Thermus aquaticus isolated by Thomas Brock from Yellowstone National Park, USA, and Wnt polymerase from Thermococcus littoralis isolated from an oceanic thermal vent. When the 3-step cycle is repeated, the 2 strands from the first cycle are copied to produce 4 fragments. These are amplified in the 3rd cycle to yield 8 double-stranded products. Thus, each cycle increases the number of target DNA molecules exponentially. Depending on the initial concentration of the template DNA and other parameters such as the G+C composition of the DNA to be amplified, it is theoretically possible to produce about 1 million copies of targeted DNA sequence after 20 cycles and as many as 1 billion after 30 cycles. Pieces ranging in size from less than 100 base pairs to several thousand base pairs in length can be amplified, and only 10 to 100 picomoles of primers are required. The concentration of target DNA can be as low as 10 raised to minus 20 to 10 raised to minus 15 molar or 1 to 10 raised to 5 DNA copies per 100 microliter. The volume of the whole reaction mixture is often 50 microliters or less. This technique was developed in 1984 by Kary Mullis at the Cetus Corporation, and he was awarded the Nobel Prize in Chemistry in 1993. PCR has revolutionized molecular biology to a very great extent and is useful in RFLP, DNA fingerprinting, detecting of mutations, bacterial and viral infections, cancer diagnosis, etc. It is also useful for amplification of DNA from very ancient and deteriorated samples for phylogenetic or evolutionary studies, or establishing hereditary DNA profiling, etc. RT-PCR is used to produce double-stranded cDNA copies of the RNA template. Gene libraries. The main purpose of genetic engineering or gene cloning is to isolate and identify a useful gene from an organism's genetic material. This is an enormous task if the gene has to be isolated each time from the original chromosome, which may be a large DNA fragment. It is like searching for a needle in a haystack. This monumental task has been made easy with the generation of gene libraries, which is a repository of genes or DNA fragments cloned into a vector and inserted in living cells or phages. There are two types of gene libraries: genomic library and Cdna or complementary DNA library. Genomic libraries. A genomic library is a set of recombinant clones that contains the entire DNA present in an individual organism, that is its chromosomes. Each clone contains a specific DNA fragment of the organism under study. That is, it is like a book in a library. Thousands of such clones represent the complete genome of the organism and hence is like a set of books in a library. The goal of genomic library construction is to have an organism's genome cloned as small fragments into separate vectors. Ideally, the entire genome is represented. That is to say, the sum of the different fragments equals the whole genome. For example, an E. coli genomic library contains all the E. coli genes, so the desired gene can be obtained by screening this library. Genomic libraries can be stored for long periods and can be exchanged between research groups. It helps to obtain specific gene for isolation and characterization. Choice of vector. The choice of vector for library construction will vary by the use and goal of the library. The most important criteria is the desired size of the insert. The following table gives the information about the various vector systems and approximate insert sizes for libraries. As shown here, conventional plasmid and phage vectors are useful in construction of gene libraries of microorganisms, as they can have inserts of about 5 to 20 kb. BACs and AACs are the more recent vectors that are useful for cloning very large, that is greater than 100 kb DNA fragments for generation of genomic libraries of higher organisms. As shown in the figure, the construction of a genomic library begins with the isolation of intact high molecular weight genomic DNA of high purity, also called Gdna, from an organism. There are several methods available for this purpose depending on whether the genomic DNA is to be isolated from bacteria, that is prokaryote, or plants and animals, that is eukaryotes. The next step is to carry out partial or complete digestion of substantial amount of genomic DNA with one or more restriction endonuclease such as BAMH1, NOT1, ZO1, etc., or by physical shearing. The DNA fragments are then inserted into a suitable vector, followed by transformation into host cells. The recombinant clones represent the library. As shown in the figure, the gDNA fragments are usually cloned in lambda based replacement vectors such as EMBL4 that can carry up to 20 kb DNA fragment. The earlier plasmid and M13 vectors were not suitable for cloning long DNA fragments. In either case, many thousands of different clones, each with a different genomic DNA insert, are created and cloned into phage particles. The next step is to screen the library with a DNA probe to select the desired clone from the library. For this, it is necessary to know about the function of the target gene or the partial or complete sequence of the gene. If the genomic library has been inserted into a microbe that expresses the foreign gene, it may be possible to assay each clone for a specific protein or phenotype. Cosmid vectors have inserts of about 45 kb. Moreover, they can replicate in E. coli and can be recovered as a plasmid. A representative gene library. A representative gene library is one that is containing at least one copy of each gene of the organism's genome. So, the question is, What should be the number of recombinant clones needed to have a representative genomic library? The following equation is used for this calculation. N is equal to log 1 minus P upon log 1 minus A upon B, where N is equal to the number of clones representing the library. P is equal to the probability that a given gene will be present. A is equal to the average size of DNA fragments inserted into the vector. B is equal to the total size of the genome. For example, if average insert size is 20 kb, probability is 0.99 and the genome of E. coli is 4.6 into 10 raised to 6 base pair and that of human genome is 3 into 10 raised to 9 base pair, the sizes of the representative genomic library would be calculated as shown. 1100 recombinant clones and 6,90,000 recombinant clones will have to be selected for construction of E. coli and human genomic library respectively. This explains that lambda vectors which can have an average insert size of 20 kb may be suitable for construction of prokaryotic genomic DNA library, but it would not be suitable for construction of human genomic DNA library as the number of clones to be handled for screening would be very large. This also causes problems for storing. This problem has been solved by the development of the new cloning vectors that can have very large DNA fragments, namely BACs and YACs. BACs are novel vectors and can have insert of about 300 kb in size, which reduces the size of human genomic library to just 30,000 clones. Isn't it desirable? Let's look into some commonly used vectors for cloning larger molecules which are useful in construction of gene libraries. BACs. As we discussed in earlier topics, bacterial artificial chromosomes was developed by Mel Simmons and co-workers in the early 1990s. BAC vectors are plasmids constructed with the replication origin of E. coli F plasmid and so can be maintained as a single copy per cell. These vectors can hold DNA fragments of up to 300 kb. BACs are often used to sequence the genome of higher organisms, plants and animals in genome projects, for example, the Human Genome Project. A short piece of the organism's DNA is amplified as an insert in BACs and then sequenced. Finally, the sequenced parts are rearranged in silico, resulting in the genomic sequence of the organism. YACs. Yeast artificial chromosomes are plasmid shuttle vectors capable of replicating and being selected in common bacterial hosts such as E. coli as well as in the budding yeast Saccharomyces cerevisiae. They are relatively small in size, approximately 12 kb, and of circular form when they are amplified or manipulated in E. coli, but are rendered linear and of very large size, that is several hundreds of kilobases when introduced as cloning vectors in yeast. YAC vectors were initially created for the cloning of large exogenous DNA segments in Saccharomyces cerevisiae, but soon became chromosomal-like platforms for a variety of in vivo experiments. Applications of YACs range from generating whole DNA library of the genomes of higher organisms to identifying essential mammalian chromosomal sequences necessary for the future construction of specialized mammalian artificial chromosomes or MACs. The availability of YAC libraries has greatly advanced the analysis of genomes previously cloned in cosmid vectors. DNA cloning into a plasmid allows the insertion of DNA fragment of about 10,000 nucleotide base pairs, but DNA cloning into a YAC allows the insertion of DNA fragments up to 1 lakh nucleotide base pairs. Other useful vector for cloning large DNA inserts includes P1 derived artificial chromosomes or PACs. PACs are vectors that combine the features of P1 vectors and BACs and have a capacity for inserts up to 300 kb. Cdna library. Complementary DNA or cDNA is derived from RNA, usually messenger RNA or mRNA. mRNA is often more abundant, that is present in multiple copies in certain cells than the usually single copy in genomic DNA. This is assuming that the gene is being expressed in the cells at that time. Therefore, it is often easier to find the gene in its mRNA form. However, if the gene is from an eukaryote, it will contain introns which are spliced out before mRNA is translated, and so the gene may be in a different form from the original DNA form. mRNA cannot be manipulated directly. Restriction endonucleases only act on double-stranded DNA, and therefore, mRNA has to be converted into DNA. This is done by using RNA as the template and the enzyme reverse transcriptase, which is obtained from retroviruses. This is the reverse of normal transcription, where the template is DNA and the product is mRNA. Synthesis of cDNA. In the first step of cDNA synthesis, a poly-DT primer is used which anneals to the 3' end of mRNAs. Reverse transcriptase then catalyzes the synthesis of a complementary DNA strand or cDNA. RNase H digests the mRNA into short pieces that are used as primers by DNA polymerase to synthesize the second DNA strand. The 5' to 3' exonuclease function of the polymerase removes all the mRNA primers except the one at the 5' end, because there is no primer upstream from this site. This RNA primer can be removed by subsequent addition of another RNase. After the double-stranded cDNA is synthesized, it can be inserted into the desired vector. Screening of gene libraries. The gene library constructed can be screened to identify the desired clone by a number of procedures. 3 common procedures are described here: colony and plaque hybridization. The principles of southern hybridization can be employed for identifying the recombinant clone containing the desired gene or DNA fragment. The first step is to transfer the colonies of plaques onto a nitrocellulose or nylon membrane. It is then treated with alkali to remove contaminants and denature the DNA molecules to generate hydrogen bonds to bind to the membranes. The cross-linking is carried out by incubating for a short period at 80° centigrade in a vacuum oven in case of nitrocellulose membrane, or by UV irradiation in case of nylon membranes. The single-stranded DNA molecules get attached to the membrane through their sugar phosphate bonds. The nitrogenous bases are free to anneal to the complementary bases in the probes. The DNA probe is labeled, denatured and added to a hybridization buffer in which the filters or membranes containing the bound DNA from colonies or plaques are present. After incubation for suitable time period, the filter is washed to remove the unbound probe, and the positive clone is detected either by autoradiography for radio-labeled probe or by a suitable detection method in case of non-radio-labeled probes. Immunological screening. This method is used when the clone DNA fragment of a gene is presumed to be expressed in the recombinant clone. That is, the recombinant clone now contains the desired protein due to gene expression. The non-recombinant host cell used for cloning the gene does not contain the protein. This is possible if pure sample of the desired protein is available and antibodies are raised against it. Anti-antibody is then prepared, which is then labeled. The recombinant colonies are transferred to a polyvinyl or nitrocellulose membrane. The cells are then lysed and the solution containing primary antibody is added. It is then detected by washing the membrane with a labeled secondary antibody that specifically binds to the primary antibody. Abundance probing to screen a cDNA library. If a genomic library is prepared from an eukaryote in an effort to isolate a structural and functional gene, a cDNA library is usually constructed. In this way, introns are not present in the cDNA library. Instead, only the protein coding regions of the genome are cloned. If a cDNA library is prepared from a tissue in which the gene is expressed at a relatively high level, then the library so constructed will contain a large proportion of clones containing the desired gene. For example, if the cDNA is prepared from germinating wheat seeds, most of the clones will be containing a copy of the mRNA transcript for gliadin gene. A cDNA clone is randomly selected from which the DNA is isolated, labeled and used as a probe to screen the cDNA library. Many cDNA clones are selected and used to screen the library. The cDNA clone that provides the probe to detect many cDNA clone in the library is the recombinant clone containing the gliadin gene and is used for further analysis. Researchers can avail gene libraries from commercial vendors or from other research labs that produce genomic or cDNA libraries of a particular organism. There are laboratories that can also generate a gene library for the organism of one's interest. So, today we have seen the use of PCR and what PCR is and how it can be used for amplifying small fragments or large fragments of DNA from different types of samples, and that can be used for various purposes. We have also seen the use of gene libraries, their construction and screening. We have also discussed the more recent vectors such as BACs, YACs and PACs that are used for cloning large DNA fragments in construction of genomic libraries. Thank you very much.